Restriction fragment length polymorphism in the human genome in between the active base pairs which code for a particular protein. There is a large number of inactive base pairs forming 95% of DNA, which is considered as a junk DNA or a filters DNA or nonsense DNA. Technically, this introns separated from the exons, which serve as a protein pattern DNA fingerprinting, overlook the DNA genome in favor of junk DNA between the gene. The junk DNA short sequence of base repeat themselves over and over again like a slutter repetitive DNA, CGTA, CGTA, GACA, GACA, etc. The regions containing repetitive DNA demonstrating hypervariability from person to person are called satellite DNA, which shows an extreme high degree of variability. This variant are called variable number of tandem repeats or mini satellite. Selected region of VND are booked into fragment using special enzyme restriction endonuclease. The resulting fragment are called restriction fragment length polymorphism (Rflp). Gel electrophoresis can be used to separate and determine the size of the restriction fragment length polymorphism. Fragment are of variable length. Exact number and size of fragment produced by a specific restriction enzyme digestion varies from individual to individual. They are individualistic in nature and establish high 100% identity. Procedure 31.1. One is blood sample, isolate DNA cut into fragments by restriction enzymes. DNA fragment separates into bands using gel electrophoresis. DNA band pattern is transformed to a nylon membrane using southern blotting and adding radioactive DNA probe. DNA probe binds to the specific DNA sequence hybridization and then it wash. Excessive DNA probe washed off. Radioactive probe is bound to the DNA pattern on the membrane. X-ray film placed to the membrane. Autoradiography of DNA pattern, DNA fingerprint, visual and computer analysis of DNA profile. Procedure: isolation and extraction of DNA first. DNA must be recovered from the cells or tissues of the body. Only a small amount of tissue, blood, hair or skin is needed. For example, amount of DNA found at the root of one hair is usually sufficient. Cutting and sizing. Special enzyme called restriction enzymes are used to cut the DNA at specific places. For example, an enzyme called E. coli 1 found in bacteria will cut DNA only when the sequence GAATTC occurs. Sorting by gel electrophoresis. The DNA pieces are sorted according to the size by sieving technique called electrophoresis. DNA pieces are passed through an agarose gel. This results in separation of DNA fragment based on the length polymorphism. This one is that mixture of DNA fragment, sample wells, buffer, cathode anode, direction of movement, plastic frame, gel, this thin semi-solid slab of agarose gel on a glass holder, buffer. Transfer of DNA to nylon southern blotting. It is possible to identify specific DNA fragment that hybridizes with a complementary genetic probe. However, it is impossible to hybridize a probe into DNA fragment contained in a gel. For this reason, DNA is usually denatured and then transferred into nitrocellulose or nylon membrane, which picks up the DNA, look like a blotter paper picks up a pink ink. DNA transferred into the membrane by capillary action and fixed by baking. It accessible to a probe. Resulting form is essentially a replica of the gel. The method of detecting DNA fragments, separating them by gel electrophoresis and then transferring them into a nitrocellulose nylon membrane is called a Southern blot, named after the inventor Dr. Edward Southern. Similar blotting technique are used to RNA northern blot and protein or peptide western blot. Hybridization, adding non-radioactive DNA probes, shorter sequence probe complementary to the regions of DNA which one wishes to dictate to the nylon sheet lead to fragment location. The nylon membrane is immersed in a solution that contains DNA probe impregnated with a radioactive P32. Each probe typically sticks to only one or two specific complementary sequence on the nylon sheet. This process is called termed as hybridization. Washing. Membrane is washed to remove excess of unbound probe, then exposed to an X-ray film. The resulting spot on the X-ray film corresponds to the location of the fragment and in gel that are complementary to the proper autoradiography. Nowadays, many radioactive probes are detected by chemical luminescence, which is analyzed by computer scanners, eliminating the need for autoradiography. DNA fingerprint. Final print is known as the autoradiograph or a DNA fingerprint, which appears as lines on the film. Current practice is use of DNA samples for crime investigation, and fraternities does not use a meticulous multilocus DNA analysis, but uses highly polymorphic single local genes such as DNTR genes. Due to the large number of distinguishable alleles in some most population, it is possible to establish a DNA signature for almost any individual. Limitations: it takes few weeks to perform. For every probing, the membrane is stripped of the previous probe and rehybridization and autoradiography performed again. It is not usable where DNA is degraded, limited value in testing cadaveric tissue for identification of human remains unless pressure. The electrophoresis technique that separates the macromolecules, either nucleic acid or protein, on the basis of the size, electric charge, or other physical properties across a span of gel motivated by an electric current. Both the DNA and RNA migrate through the gel towards a positive pole of the electrical field because they are negatively charged due to their phosphate group. Okay, so this one is the isolation and extraction of the DNA, cutting and sizing by using an enzyme, restriction enzyme, sorting by gel electrophoresis, transfer the DNA to nylon southern protein, and hybridization by adding radioactive DNA probe, then DNA fingerprinting. Thank you.